closely at a drop of pond water, and you find a dense, overlapping tangle of cellular shapes and Latin binomials. The sheer, unorganized diversity of prokaryotic life is staggering. Biologists manage this density using a strict 5-volume phylogenetic framework. Historically, this architecture has been codified by Bergey's Manual of Systematic Bacteriology. This handwritten layout maps out the highest levels of that exact system. We are going to trace these raw taxonomic notes to build a working mental model of how microscopic life is actually categorized. First, we organize organisms based on their most extreme metabolic capabilities. Second, we map the massive internal diversity of the gram-negative proteobacteria matrix. Third, we split the gram-positive bacteria across a deep genetic divide governed by their DNA composition. Finally, we isolate the structural and morphological outliers that defy standard grouping. By mastering this 5-volume architecture, you convert raw biological chaos into a highly navigable, predictive scientific tool. Volume 1 serves as the repository for the deepest phylogenetic branches. This includes the entire domain Archaea, alongside the most ancient, deeply branching lineages of domain bacteria. Looking at this top section of the framework, domain Archaea is primarily divided into two main phyla, the Crenarchaeota and the Euryarchaeota. Classification within the Euryarchaeota relies heavily on an organism's adaptations to extreme environments and their specific metabolic outputs, particularly in anaerobic conditions. For example, this section details Class 1, the Methanobacteria, strict anaerobes whose end product is methane. Moving down to Class 4, the Halobacteria present a strict contrast. Their taxonomy requires high salinity, noted here as needing 1.5 molar sodium chloride for growth. Crossing over to domain bacteria within this same volume, we find the deeply branching phototropes. Rather than salt or methane, these bacterial lineages are categorized strictly by their relationship with oxygen during photosynthesis. Down here, the notes on phylum Cyanobacteria specify that they are oxygenic. They utilize a complex photosynthetic apparatus remarkably similar to eukaryotes. Just beneath that, phylum Chlorobi, the green sulfur bacteria, are explicitly marked as anoxygenic. They perform photosynthesis without producing oxygen at all. At the deepest roots of the phylogenetic tree, the primary taxonomic boundaries are drawn by the mechanics of metabolism, what an organism produces, and the extreme conditions where it thrives. Volume 2 contains the proteobacteria. This is the largest, most diverse phylum of gram-negative bacteria in existence. To manage this overwhelming diversity, the phylum is mapped across a clean organizational grid of 5 classes, designated by the Greek letters alpha through epsilon. This page documents that classification, starting with Class 1, the alpha proteobacteria. Here, Rickettsia is characterized as an obligate intracellular parasite. Below that is Class 2, the beta proteobacteria, containing major environmental actors and human pathogens like Neisseria, noted as the causative agent for meningitis and gonorrhea. The third class, the gamma proteobacteria, represents the most economically and clinically significant group within the entire phylum. The variety in this Class 3 list is massive. It ranges from food spoilage agents like Pseudomonas to critical enteric microbes like Escherichia and Salmonella. The clinical impact is severe. Notice the inclusion of Yersinia, annotated directly with the historical disease it causes, plague. Class 4, the delta proteobacteria, shifts away from human pathogens to focus on unique environmental behaviors and roles within the sulfur cycle. Look closely at this boxed entry for Bdellovibrio in Class 4. It is classified by its highly specialized lifestyle as a predator that actively hunts other gram-negative bacteria. The tour of the proteobacteria concludes with Class 5, the epsilon proteobacteria, featuring distinct specialized pathogens like Helicobacter. Despite ranging from intracellular parasites to predatory hunters, every organism in these 5 classes is grouped together based on a shared gram-negative evolutionary lineage. Volume 2 functions as a strictly organized matrix for cataloging the most impactful gram-negative bacteria in science and medicine. The architecture shifts entirely when classifying gram-positive bacteria, which are defined physically by their thick peptidoglycan cell walls. This diagram illustrates the core dividing line for gram-positives. It is entirely genetic. Taxonomy here relies on the G+C ratio, the percentage of guanine and cytosine base pairs forming dense triple-bonded connections in the organism's DNA. Volume 3 of the manual is dedicated to the Firmicutes. These represent the low G+C gram-positive bacteria. Volume 3 covers Class 1, Clostridia, which form endospores in strictly anaerobic environments. Panning to Class 3, Bacilli, we find pathogens like Listeria, linked to food poisoning. Class 2's Mollicutes lack a cell wall, yet their genetic lineage proves they are phylogenetically gram-positive. Organisms with a higher density of those triple-bonded base pairs are separated into Volume 4, the Actinobacteria, representing the high G+C gram-positives. On this page detailing Volume 4, Mycobacterium stands out. It is classified here by the high lipid content in its cell walls, which results in its unique acid-fast staining properties. This volume also captures highly complex, branching bacteria like Streptomyces, which are critical to industrial medicine and antibiotic production. For gram-positive organisms, observable physical structures are secondary. Deep genetic composition determines evolutionary relationships. Volume 5 serves as the taxonomic home for the remaining phyla. These are the organisms that do not fit into the Proteobacteria matrix or either side of the gram-positive genetic divide. Many groups here are defined by highly unique cellular architectures, like the internal axial filaments that allow this spirochete to rapidly corkscrew through viscous fluids. This final list outlines those distinct phyla, specifically detailing the Chlamydiae and the spirochetes. The clinical significance of the spirochetes relies on the identification of specific genera listed here, such as Borrelia and Treponema. Other highly distinct groups, including the Fusobacteria and the Verrucomicrobia, round out the remaining entries. Volume 5 collects the structural extremes of the bacterial world. Here, unique morphology dictates an organism's phylogenetic placement. When we expand these isolated details into a single, fully populated network, the unified architecture of the taxonomic system becomes clear. This structure operates as a predictive engine. Identifying which volume an organism belongs to immediately signals its likely metabolic pathways, genetic makeup, and clinical behavior. If researchers isolate a newly discovered high G+C bacterium, the taxonomy predicts it will share resilient, complex traits with known genera like Actinomyces. Similarly, identifying a new JAMA proteobacterium instantly warns of a gram-negative organism with high pathogenic potential. By systematically mapping extremophiles, genetic ratios, and unique morphologies, we convert biological chaos into a navigable, predictive database for modern science.